Hello everyone, I am Dr. Reena Mehta and I'm an allergist and immunologist at Uptown Allergy and Asthma in New Orleans, Louisiana. I'm excited to talk to you today about COVID-19 vaccines. I do not have any relevant disclosures. So today we'll be talking about mRNA vaccines as a whole. We'll be talking about the approved COVID-19 vaccines, those that are also in the pipeline, as well as mutant strains and vaccine strategies. So here is a quick refresher on the different kinds of vaccine platforms and how they relate to current COVID-19 vaccines. We have DNA and RNA-based vaccines. We have inactivated vaccines, viral vector vaccines, and subunit vaccines. So just to remind everybody how mRNA COVID-19 vaccines work. mRNA vaccines give instructions to our cells in your body to make a spike protein or a small piece of the COVID-19 virus. That is then injected to allow the immune system to create protective antibodies. So it's important to note that mRNA vaccines do not affect our DNA because they do not enter the cell nucleus. And while these our first available mRNA vaccines is for COVID, this is a technology that is not new and has been studied for a long time. So just to bring you back to the immunology of how these mRNA vaccines work. So the mRNA is transcribed in vitro, which then enters our cell cytoplasm through endocytosis. This is then presented to the ribosome, which then makes its way to the proteasome for degradation into antigenic peptides, which are then loaded onto our MHC class I molecules and presented to the cell surface for a CD8 T cell response. Also, this can be done with antigenic peptides presented to MHC class II molecules, also going to the cell surface to create CD4 T cell responses. So there are a few advantages of mRNA vaccines over our traditional vaccines because they are safe, efficacious, and induce both B and T cell responses. mRNA vaccines do not form viral particles, so there is no risk of inducing disease. And mRNA vaccines do not integrate with our DNA or enter the cell nucleus. mRNA vaccines can be mass-produced in large quantities rapidly. Since viral particles are not used, their production is easy, safe, and cost-effective. mRNA vaccines are typically preferred over DNA vaccines because the production of DNA vaccines is cumbersome since genetic information needs to enter the nucleus. What are some potential disadvantages of mRNA vaccines? Free or naked mRNA molecules are unstable and transient. Thus, mRNA needs to be incorporated into a lipid layer to make it stable, which is what we've been able to see with the COVID vaccines. So the COVID pandemic has led to rapid development of vaccines. While most vaccines take years to even get to phase 3 trials, we have gotten there impressively in less than a year. And so I just want to make note that today is February 9th, 2021, and when I'm recording this lecture, and so you all are viewing it in a few weeks from now, so a lot of things may change. Our literature and the studies are rapidly changing, so I just wanted to give you, today is what we know as of February 9th. So let's talk about our COVID-19 vaccines. Currently, there are 4 which are approved for full use and 6 that are in early or limited use. So now we'll go into detail about all of the different COVID-19 vaccines. There's many, but we're going to focus on the ones that are in phase 3 trials, available for use, or most studied. So first we'll talk about mRNA and DNA-based vaccines. So there are only 2 vaccines available in the United States currently under emergency authorization, and both are mRNA vaccines. I would assume that the majority of you have already received one of these vaccines, thus I will not go into too much detail about the different phase 3 trials, as I'm sure you know a lot of this information. The key facts that I would like to point out is that both are over 90% effective in preventing COVID infection. Both are given in 2 doses about 3 to 4 weeks apart. The Pfizer vaccine needs to be stored at specific freezer temperatures, and they are now looking at the safety in adolescents through certain trials. CureVac is an mRNA vaccine in phase 3 trials in Germany. Angus is a DNA-based vaccine in phase 2-3 trials in Japan. Zy-CoV-D is another DNA-based vaccine which is delivered through a skin patch, which is in phase 3 trials in India. The efficacy is still unknown. Now that we have covered mRNA and DNA-based vaccines, we will discuss viral vector vaccines where a weakened version of a virus, such as adenovirus, serves as a vector and carries a gene for another virus, in this case, SARS-CoV-2. Sputnik V is a vaccine which uses adenovirus as its viral vector. It has a high efficacy, over 90%, and is widely used in Russia. Currently, there are phase 1 trials looking at whether this vaccine can be given in 1 dose rather than 2. Here is the phase 3 trial and the results of the Sputnik V vaccine. It was published in Lancet. They enrolled over about 22,000 participants and found over a 90% efficacy in the vaccine group. Another viral vector vaccine is Covishield, made by AstraZeneca, which uses chimpanzee adenovirus vectors. This vaccine has been widely used in the UK, India, and Mexico and shows on average about a 70% efficacy. This phase 3 trial was also published in Lancet, which includes over 11,000 participants, and the vaccine was found to be about 70% effective. What was interesting about this trial was that there was a subset of participants who received a half dose for the first injection. The results were surprising because the group that received the half dose on the first injection actually had a better efficacy than those that received the full dose, which raises the question, did the lower first dose mimic actual infection, promoting a stronger immune response? Currently, the U.S. is in a large phase 3 trial where with this vaccine, so AstraZeneca may be applying for emergency authorization in spring of 2021. AstraZeneca also just reported that increasing the interval between the first and second dose from 4 weeks to 12 weeks leads to an improvement in efficacy. This may be helpful in areas where there is vaccine shortage to give more time to secure a second dose for people. There is a new trial starting in Ukraine to see whether combining the AstraZeneca with Sputnik V will increase the efficacy of the vaccine. These results will be interesting when they're available. This brings us to the next major viral vaccine from Johnson & Johnson, which is also adenovirus-based. This vaccine is given as 1 dose, which is different than the other COVID vaccines we have discussed so far. In the phase 3 studies, the efficacy has varied depending on where the vaccine was administered, meaning location, with a lower efficacy in South Africa, probably due to COVID-19 mutant strains, which we'll talk about soon. However, this vaccine did protect everyone from about 80% from severe infection. The U.S. applied to the FDA for emergency authorization for this Johnson & Johnson vaccine on February 4th, and the FDA will meet to discuss this on February 26th. There's also another phase 3 trial looking at giving this vaccine in 2 doses and whether this will improve its efficacy. So stay tuned on results from that. Another viral vector vaccine is Canvassidia, which is in limited use in China. Now we will move on to protein or subunit-based vaccines. The best known subunit vaccine is Novavax, which has an efficacy of almost 90%. Given its lower efficacy in South Africa, they are developing a new version tailored to this variant. In the United States, phase 3 trials are currently underway with results expected in March. Epivaccia is another vaccine in early use in Russia. Next up, we're going to talk about inactivated COVID vaccines. Sinopharm is one that is used in China and other parts of the world with about an 80% efficacy, but their phase 3 trials have not been published yet. Covaxin is approved for emergency use in India. Okay, so now that we have discussed the various types of COVID-19 vaccines, those that are approved and those that are in the pipeline, now let's talk about what we're all worried about is these mutant COVID-19 strains. So as you can see here, the mutant actually comes due to a variation in the spike protein. There have been 3 major variants that have been studied, although there are more reported every day in the news. The first variant that came to be known was in April-May, the D614G. Unfortunately, studies have shown that the various vaccines are effective against this strain. The 2 more worrisome strains, the B117 from the United Kingdom, otherwise known as the UK variant strain, and the South African variant, have been shown to cause more severe illness with concerns about loss of vaccine efficacy. The concern is that these mutations in the S protein can increase the rates of transmission, increase the amount of virus that is shed, and can also compromise vaccine efficacy due to destroying areas on the neutralizing antibody receptor site. There have been a few non-peer-reviewed studies looking at these variants and vaccine efficacy. One small study included volunteers who received either Moderna or Pfizer, and they found that the South African strain modestly reduced the efficacy of these vaccines. Taking it one step further, Moderna released a study that looked at serum from some of their phase 1 participants comparing antibody production against the different variants. They found that all participants produced an adequate antibody response to the D614G variant and the UK variant, but there was a 6-fold reduction in protective antibodies to the South African variant. Pfizer then published a small study looking at participants in their phase 3 trial and found that all patients produced neutralizing antibodies to all COVID-19 variant strains. However, this study was limited because it did not include the full set of spike mutations in the UK and South African variants. Thus, it remains unclear whether the reduction in antibody response to the South African strain is enough to reduce vaccine efficacy and if this will translate into more people getting sick despite vaccination. mRNA vaccines have the benefit of producing T cell responses, so maybe antibody response is not the only way to be protected. Also, mRNA vaccines induce such a strong neutralizing antibody response, as seen with 90% efficacy in our Pfizer and Moderna trials, that maybe a little reduction may not be clinically significant. This is all interesting things, yet we still don't know. What these variants should do is raise a red flag. We need to continue to vaccinate the population at large to prevent the emergence of more variants. We need to test more people for these variants, and new vaccine efforts must be made to protect against these variant strains. The good news is that the mRNA platform has a lot of flexibility and can be easily and quickly modified to adjust for new mutant strains. And we hope that with both B and T cell responses by mRNA vaccines, we will still maintain an adequate protection against a majority of these variants. So what is in the future? Moderna will launch 2 trials, one to see if a 3rd booster dose could provide further neutralizing antibodies, and 2, Moderna is developing a booster vaccine against the South African variant, and phase 1 trials are underway in the United States. What can we do to make our mRNA technology even more convenient in the future? We can try to create a more potent vaccine that only needs to be given in a single dose. We can try to create vaccines that are thermostable so that they do not need to be refrigerated to make distribution easier. It is still too early to know how other non-mRNA vaccines will be affected by these variant strains. But based on a small study which showed low efficacy against the South African strain, they have halted distribution of the AstraZeneca vaccine in South Africa. So there's still a lot of unknowns, but we've come a long way in a year's time in terms of, trying to fight this pandemic and getting, really efficacious vaccines out there for public use. And so in conclusion, the COVID-19 pandemic has led to accelerated preparation of suitable COVID-19 vaccines. mRNA vaccines are safe, efficacious, do not integrate into our host DNA, and can be manufactured and altered rapidly. Currently, there are 2 approved mRNA vaccines for emergency use in the United States, Moderna and Pfizer. And viral vector vaccines, such as J&J and AstraZeneca, and peptide-based vaccines, such as Novavax, are efficacious and will likely be available in early spring of 2021 for emergency use. Mutant COVID-19 variant strains are concerning and may cause more severe disease and evade vaccine immunity. The most concerning one at this time is the South African variant. Booster dosing of current COVID-19 vaccines may help provide protection against viral mutant strains. So with that, I will end. Unfortunately, you're not able to ask me questions since this is pre-recorded, but if you want to talk further about COVID-19 vaccines or have any questions, all of my contact information is listed here. Please feel free to reach out anytime, and I hope you all have a wonderful rest of your time with the conference. Thank you so much for your time.